Hey guys, today we're talking about ELISA, which is also known as enzyme-linked immunosorbent assay. There are many uses for ELISA, and basically this just tells you and informs you about if there's any specific foreign antigens within your body. So in our case, we're tracking down basically Neisseria meningitidis, which is basically a bacteria that targets the nervous system, and basically this bacteria can be found within your spinal fluid. Here we have a diagram of what Neisseria meningitidis looks like, and the little green dots on top of the surface of the membrane is known as the antigen. So now if we get a closer look at our antigen, this is what it should look like. So our antigen, basically for each specific antigen has an epitope. What our B cells do is that they create specific antibodies. So one single antibody that is made for a specific single epitope on one antigen. Of course, for them to bind, they should fit perfectly. So when testing whether if a patient has this specific antigen or the condition, we basically want to get a sample from them, whether this could be a bodily fluid. For Neisseria meningitidis specifically, we should definitely use a spinal tap in order to get cerebral spinal fluid, because the bacteria itself targets the nervous system. Basically, our antigens will actually stick to the plastic, and then we would want to wash them. We add in the antibody made for specifically this antigen. So again, like this formation, these two will attach. Alright, so now that they have attached, we would want to give them a wash to cancel out any background noise that might affect the results. This specific formation is also considered to be called monoclonal, and make sure this is from one specific animal. Our specific situation is to be a type of ELISA known as indirect ELISA. We must add secondary antibodies. These secondary antibodies will basically be derived from another animal, and they are also known to be called polyclonal. We use secondary antibodies because these actually amplify the signal of the antigen. After that, we will add in the horseradish peroxidase, which is also known as an enzyme that will attach to our secondary antibodies. Of course, we know that we can't have an enzyme without our substrate, so we are going to add in our TMB, also known as tetramethylbenzidine. Of course, after our enzyme and substrates bind, will be indicated with a color change. Color change will indicate whether our patient has Neisseria meningitidis or not.